Because they like to survive into adulthood, and in order to do that, they have to avoid being eaten by their predator. If you can't recognize your predator, it's a lot harder to avoid being eaten by your predator. If you don't know when to use those anti-predator behaviors, you might not use them at the right time and end up getting consumed by somebody bigger.
The embryo stage of life is a very vulnerable stage for these fish. They don't have a lot of ability to escape a potential predator coming around. Really, all they can do is potentially avoid detection.
Our research mostly focuses on detection via olfactory cues or scent-based cues. So essentially, we think that they smell their predators. There is research from other labs into other things like visual cues or tactile cues if they get poked or anything like that, but our research is just with those smell-based cues. There aren't a whole lot of anti-predator behaviors available to you when you're still stuck inside an eggshell. In science, we call that the chorion, so that's a fun vocab word for you. The eggshell is the chorion. The main behavior that we look for at this point is freezing. So normally, these baby fish still inside their chorion would be wiggling around and moving and rolling over, doing all kinds of little movements. But if they smell a predator, they'll hold still, they'll freeze, and wait until that cue is gone and the danger has passed.
It's adaptive for embryos to also be able to learn to recognize a predator before they're hatching. Adaptive means that it is something that is going to be beneficial to the embryos in terms of increasing their survival. During the vulnerable embryonic stage of life, an embryo really doesn't have a lot of options available to it to avoid a predator that's in the area. It can't run away, it can't avoid that predator in any way. The only thing it can really do is try to minimize detection by the predator, perhaps by getting very still. In order to be able to know when they should do this, they have to be able to learn to recognize when there's a potential threat in the area. That's why it's important for embryos to be able to learn to recognize predators.
Their eggshell is semi-permeable, which means that it's not a completely hard barrier to their environment. So the eggshell is transparent, so they can see potentially visual cues. And it's also, because it's semi-permeable, like I said, it's a texture that allows for certain smells or water to pass back and forth between the inside and the outside of the egg. So they're still able to detect scents even though they're still trapped inside this egg.
The alarm cue is a component of the skin cells in minnows. So the way we get the alarm cue is by removing the skins from minnow fish and extracting the alarm cue by crushing those cells. So we keep a colony of sunfish here at the lab, and the way we collect cues from these sunfish is that we collect the water that they're living in. So basically, our predator cue is their pee. One of the things that we noticed is that if you pair an alarm cue with a predator cue, they will learn the association between the alarm cue and the predator cue. And they will recognize that predator cue as being a potential danger. Then, if you give them the predator cue alone, later on, they will respond to that predator cue with the recognition that this is a dangerous situation. The way that we observe that happening in minnows is that within the egg, their behavior changes. Normally, these embryos are flipping around, they're twitching within the chorion, there's a lot of spontaneous activity, but those that have been taught to recognize the scent of a predator will show a change in their behavior when they receive this predator cue. And usually, they will start to get very still and get very quiet within the egg and try to avoid detection. Fish can certainly learn to recognize those same signals after they hatch as well. So fish, after they've hatched, will show a similar ability to learn to pair any olfactory scent in the environment with the alarm cue and begin to perceive that new scent as being a danger and respond to it accordingly. Fish, after they're hatching, definitely learn from watching what other fish do. Younger fish will learn from older fish, but even just in fish shoals, fish watch what each other are doing, and then they will learn what is dangerous, they'll learn the position of new food resources, and they'll learn about other aspects of the environment. There's a lot of social learning that goes on in fish populations. One of the things that we're really interested in knowing is whether or not embryos also exhibit that form of social learning before they hatch out. This is something we're actively investigating right now, and our early results suggest that certainly embryos do pay attention to what other embryos are doing. So when we're testing embryos, we put them under the microscope, and then we can present them with different cues. Some of those are predator cues, and then we can observe how their behavior changes. What we're looking to see is how their spontaneous movement within the chorion changes in response to the olfactory sense that we're giving them. So when we record these data, we can subject it to an automated tracking software that will record the amount of time that the embryo is being active within the egg, the number of flips and turns that the embryo does, the amount of time that it spends inactive in the egg, and when during the test did it do all of these things. The software automatically will track the behavior of multiple embryos at a time, which is really beneficial to us because the sample sizes that we use are in the thousands. So we will normally test thousands upon thousands of embryos for any particular experiment. It would be very time consuming to try to manually measure what all of these embryos are doing one by one.